Welcome to this Oncology Data Advisor interview. Today we're having this discussion in honor of Esophageal Cancer Awareness Month in April, and I'm joined by Dr. Harry Yoon, who is a professor of oncology at the Mayo Clinic. Dr. Yoon was the lead investigator of the Rationale 306 trial, which recently led to the FDA approval of Tislelizumab as first-line treatment for unresectable or metastatic esophageal squamous cell carcinoma. And today, Dr. Yoon is here today to tell us more about this approval and insights for the Awareness Month. So, Dr. Yoon, great to have you on the show today. To start off, would you like to introduce yourself and share a little bit about what you do?
Sure, pleasure to be here. I'm a GI medical oncologist and clinical investigator based at Mayo in Minnesota, and I help lead the research, practice, and education activities across the three Mayo sites across the enterprise in the country. And I have a NIH-funded lab, and we're trying to understand how to enhance the cooperativity between immunotherapy and angiogenesis therapy.
Awesome. Excited to talk to you some more today. So, for background, before we delve into this approval, could you give us a little bit of an overview of the challenges surrounding esophageal squamous cell carcinoma and the need for more treatments?
Sure, this is one of the most common cancers in the world, and a very aggressive cancer. Median survival has been only about a year in patients with stage 4 disease, which is a large portion of this population. So clearly an area of unmet need.
So, looking forward to hearing more about tislelizumab today. Would you like to tell us what kind of treatment is this and what is its mechanism of action?
Sure. So Tisla is a PD-1 inhibitor. It's very much like Pembro or Nivo.
So, would you like to tell us some more about the Rationale 306 trial, what was studied in it, and then what results the trial produced?
Sure. So the trial Rationale 306 was a global phase 3, and it tested whether adding Tisla to standard chemo could improve overall survival. And it was a placebo-controlled trial. So Tisla was compared directly with placebo, and both arms got chemotherapy. So the trial met its primary endpoint, which was overall survival, and the median survival was about 17 months with Tisla versus 10 months with placebo. And something that was notable was that 3 years later, with a minimum of 3 years of follow-up, about 15% of patients in the Tisla arm remained progression-free. So about 15% still did not have disease progression 3 years after. So this is with metastatic or advanced esophageal squamous patients. And so it's a really remarkable outcome in patients with this type of disease.
It's really exciting to hear. Thanks for sharing these results. So now that Tisla is approved for first-line treatment, what does this approval mean for patients? How do you see it fitting into the treatment landscape for esophageal squamous cell carcinoma?
So I think the big picture is that it gives another option for patients in the US that's at least as efficacious as the two other options that are available to us, which is right now Pembro and Nivolumab. And then the more specific question, in terms of where does Tisla fit? If I'm a practicing oncologist, how could I think about it? And so in the US, we like to give immunotherapy with Folfox. So nivolumab works well in that setting in terms of synchronizing schedules because they're both given typically every 2 weeks. Another option is Pembro, which can also be given every 6 weeks, and that tends to work well. So one thing that distinguishes Tisla from Pembro and Nivo is that Tisla is the only immunotherapy in this space that was tested in a global phase 3 in combination with a taxane doublet. Both Pembro and Nivo were only tested in combination with a platinum fluoropyrimidine in this particular setting. So the most evidence-based option would be to use Tisla in situations where a fluoropyrimidine is not the preferred chemo backbone. So let me just say that last word. So the most evidence-based option would be to use Tisla in situations where a fluoropyrimidine is not the preferred chemo backbone. And so some examples of that could be patients who might wish to avoid a 46-hour 5-FU pump because this can be cumbersome for some patients, or patients who have a DPYD deficiency. And some medical centers are moving into testing for DPYD more proactively. There's also some people might want to use capecitabine, at least in other countries, which allows for a every 3 week schedule. But CAPE is usually not a viable option for first-line esophageal cancer patients because they typically have significant issues with dysphagia.
It's really great to know about this new option. So what should clinicians know about adverse event management as they begin prescribing this medication?
So the side effect profile for Tisla is very similar to that of the other PD-1, anti-PD-L1 inhibitors, and there's no new safety signals.
It's great to know. So looking ahead, what are some of the other advances that you're looking forward to seeing in the treatment of esophageal cancer in the coming years?
So I think, that there's a few things. What studies like the Rationale 306 and others are showing is the promise of targeting adaptive immunity, particularly with immune checkpoints that serve to regulate the cytotoxic T cells. So there are several other immune checkpoints that are being targeted, and I think those are worth, investigating. Another promising avenue is actually kind of going backwards in a way, because we've been going so much in the direction of biologics, but going backwards in a sense to enhance our understanding of the importance of traditional cytotoxic agents, so to speak, or cytotoxic payloads, and especially how to increase their target specific delivery deep into the tumor tissue. That's been a major issue. And this is important because so far, very few patients can have a durable tumor control on biologic therapy alone. So I think one area that is showing a lot of promising results are antibody-drug conjugates or ADCs, where the conjugated cytotoxic payload can presumably better reach the target tumor cells compared with free payload, compared with your traditional cytotoxic agent just delivered by itself. And these ADCs might be able to kill more tumor cells also through bystander effect, killing the neighboring tumor cells, even though those neighboring tumor cells might not carry the biomarker, might be biomarker negative, or they might carry the biomarker, but only at a low expression level. So I think, those are two areas. A third area, the last one I might mention right now, of kind of high interest and also overlaps with my personal interest is whether targeting angiogenesis can be combined with PD-1 blockade in this disease. And there's some, I think, intriguing data that's coming on this in the future.
Awesome. It'd be exciting to see these new advances as they fold out. So my last question for you today is, since April is Esophageal Cancer Awareness Month, are there any messages you'd like to share with our audience to raise awareness of this disease?
Yeah, this is a great question. I think the big thing is the death burden of esophageal cancer. Esophageal cancers are the sixth most common cause of death in the world. There's more than 500,000 deaths per year. So clearly an area of unmet need. And even with these immunotherapy advances, patients, most of them are still progressing at some point or have to stop therapy because of toxicity. But, these are really promising advances. And now, remember before I said the median overall survival was typically less than 12 months, and now we're approaching the 1.5 year mark, at least for a trial eligible patient. So, because of the high death burden, this is still far from where we want to be. So continued public support for research, I think, is key to build on these advances.
Great. It's a great message to wrap up on to help raise awareness this month. So thank you so much, Dr. Yoon, for coming on today to tell us all about this trial and the approval and sharing your insights today.
Great, and it was a pleasure to be here.